Since joining Beyond Nuclear in 2007, Cindy Folkers has focused on ionizing radiation and its impact on health and the environment, both from a scientific and historical perspective. She advocates for fully protecting the health of females and early life stages in the face of uncertain health outcomes. She earned an MS in environmental science from Johns Hopkins University. She investigates radiation science and the historical context which informs that science, noting where research conclusions often vary greatly from the lived experiences of exposed communities. She has worked with frontline communities, including those around uranium mines and nuclear technology facilities, to understand why their actual experience differs from official research and regulatory expectations. She analyzes radiation exposure policies and advocates for scientific and regulatory methods that can close the gaps between actual health impacts and the science and regulation that are supposed to represent these impacts, and encourages communities to self-advocate for this result as well, with a focus on cutting-edge scientific techniques such as radiation bioindicator and genetic research. In addition to communicating with frontline communities, she also engages with media, members of the public, US Congress members, and national and international agencies. She emphasizes the use of precaution in radiation exposure and advocates for public openness regarding contamination monitoring, the science surrounding determination of harm, and the basis for exposure regulations. I first met Cindy about 2 to 3 years ago, the night before Helen Caldicott was honored at the PSR National Meeting in Washington, DC. We talked well into the night. Cindy is my expert go-to encyclopedic academic researcher on all things related to the human impact of ionizing radiation. I am grateful for her expertise and so thankful that all of you will also get to learn from her now.
Good afternoon. I'm Cindy Folkers, and I'd like to thank you for coming to my talk today. And I'd like to thank PSR Colorado for asking me to speak and for hosting this symposium.
We carry the stories of radiation exposure in our bodies, but we have to know how to read them. You've heard a lot at this conference on contamination at and around Rocky Flats, and I'm going to present a more universal, historical, and health context, including how I see biomarker research fitting in. Right now, there remains a disconnect between the lived experience of exposed communities and official proclamations that health damage at lower and chronic doses is minimal. Independent science, on the other hand, has often better reflected the health experiences under these circumstances. Biomarkers in particular are important because disproportionate impacts on childhood and pregnancy have been minimized historically, and bioindicator science may help clarify exposure damage to people around weapon sites like Rocky Flats, but also civilian nuclear facilities. Radionuclides can accumulate in our bodies, and these levels can be measured. But radionuclides don't just collect in our bodies. Their radioactivity can often leave traces behind. These traces, in the form of malformations, can also be observed and measured. In a number of cases, these malformations are not only associated with exposure to radioactivity, but also with increased disease risk from that exposure. Often it's malformations in chromosomes, which is genetic material, and cell nuclei, and you can see visual representations of those malformations on this slide. The malformations I'm showing you all start with a chromosome break, and then a recombination or reformation of the now broken genetic material to a mutated form, meaning all of these formations can go on to initiate disease. Translocations are associated with causation of blood cancers like leukemia. Micronuclei shown here are chromosomes or parts of chromosomes that have not been incorporated properly into the cell nucleus. They can play an active role in tumor biology. Dicentrics have a role in cancer initiation and progression, and these are just a few examples. Some biomarkers can be caused by exposures other than radiation, and many more biomarkers than these ones exist. I call these biomarkers sort of the classic ones. One can argue that there are a number of beginnings to the nuclear age. The first atomic bomb explosion took place over Trinity in New Mexico, and it would certainly not be the last. The gadget exploded in 1945 in an area known as Jornada del Muerto, roughly translated as the Walk of the Dead Man. People who were being exposed to ionizing radiation long before this, but it was less widespread. Following Trinity would be a series of atomic explosions that would expose people worldwide and leave a legacy of contamination we still carry in our bodies. And you can see this map, how far the radioactive fallout from Trinity reached. It crossed state lines. And this graph shows a declining rate for infant mortality until 1945, when the trend was broken with this spike, presaging the increased sensitivity of early life, that's pregnancy, infancy, and childhood, to radiation exposure. While publicly denying health data from events like Trinity and accusing those who were concerned about health impacts of being radiophobic, nuclear proponents in the government had started a series of secret, classified radiation experiments. Many of these experiments were designed to measure the amount of radiation in the human body from bomb test fallout, and a number of them were done on humans without their knowledge or consent. You can see here original records from Project Sunshine, and a record of collection of body parts from deceased fetuses, babies, and children, an undertaking dubbed body snatching by one of its architects, Atomic Energy Commission employee Willard Libby. It was conducted worldwide and without the knowledge or consent of the parents. And in the case of the Atomic Energy Commission, they weren't asking the question, how much does a certain level of a radionuclide damage living beings? The question instead was, how much radiation can we expose people to before any diseases become noticeably associated with it? This would be the basis for establishing an acceptable injury limit that would be necessary for the nuclear industry to proceed. This dose is today known as a permissible dose, the word injury being scrubbed out. But merely finding the presence of radionuclides, in this case strontium-90, a bone seeker, only presents a part of the picture. What biomarkers allow us to do is see damage from radiation exposure at ever smaller doses, helping us understand that damage is occurring, which could result in disease, even if we don't see disease right away. In the center here is physician Louise Rees. In 1959, Rees and her husband Eric joined environmental scientist Barry Commoner, pictured here in the glasses, to collect baby teeth. They did so after biochemist Herman Kalkar published a 1958 article in Nature entitled An International Milk Teeth Radiation Census. They ground these teeth looking for strontium-90, which was quite a bit less gruesome than stealing the bones of dead children. They found that children born in 1963 had absorbed levels of strontium-90 that were 50 times higher than those found in children born a decade earlier. And it was at this time that we see not only biomonitoring techniques being used, but also use of biomarkers. And I will now highlight 4 different studies that have used biomarker techniques. Weaponeers weren't just collecting information from our bodies about how much radionuclide was collected there, but they were using malformations in cells to discern how we may be suffering damage from radiation exposure. By examining radium dial painters who had been exposed decades earlier and their offspring. These mostly young women, a large number still teenagers, had been exposed to large doses of radium through inhalation and ingestion as they licked their paintbrushes before painting numerals on watch and airplane dials. The radioactive radium paint would allow the dials to glow in the dark. In this 1963 study, the dial painters and their offspring were examined using cytogenetic techniques. This diagram shows 3 generations. The first is the dial painter. In this case, it was 2 sisters. And rows 2 and 3 are 2 generations of offspring. Now, I show you this not to comment on either the impact of the dial painter's exposure on their offspring or how well the study was conducted, but to illustrate the use of cytogenetics occurred fairly early on in the bomb age. And aberrations found in this cohort included a ring chromosome, aneuploidy, isochromatic breaks, no dicentrics. Abnormalities in the offspring were similar to those found in the dial painters themselves. And the radium dial painters, by anyone's estimation, have huge doses of radium exposure. But when you're dealing with radiation, large doses can be imparted by vanishingly small amounts of material, particularly when you bring it into your body through inhalation or ingestion. Later use of cytogenetic techniques would investigate doses much lower than this. At the 3 Mile Island civilian nuclear facility in Pennsylvania, the 1979 meltdown of one of the cores released enough radiation to peg the meters. That means the meters were flooded with so much radiation that they couldn't actually measure it anymore. 15 years after these releases, 29 people who experienced erythema, which is skin reddening, vomiting, and diarrhea, had their blood drawn and examined for dicentrics. From this analysis, it was concluded that these 29 people had sustained doses between 600 to 900 milligray, that's 60 to 90 rad, around the time of the 3 Mile Island meltdown. The officially accepted doses were much lower than this. In 2019, I stirred a debate when I again mentioned this study in the presence of National Cancer Institute researchers. As a result, there were published papers, and a debate continues now concerned with how long dicentrics and the cells that carry them actually last. Before this, these NCI researchers didn't know of this study's existence or that it had made use of dicentrics, despite repeated attempts and from numerous people to actually raise it since the study's publication in 1997. Chernobyl exploded in 1986, releasing hundreds of radionuclides Europe-wide and over the former Soviet states. Fallout reached the US West Coast, where there were unexplained bird deaths at the time. 55 adults and 25 children who were 100 to 200 kilometers from the Chernobyl explosion in 1986 had their blood drawn and examined, looking for micronuclei. This was after they immigrated to the US between 1989 and 1991. A whole body counter was used to determine internal radiocesium contamination. The presence of micronuclei was significantly and positively associated with the internal contamination level of radiocesium, and the estimated internal absorbed dose ranged from 0.6 milligray, that's 60 millirad, to 9.2 milligray. It was not expected that the incorporated radiocesium exposure was the only cause of micronuclei, as they had also suffered external exposures, But as I will point out later, the concept of a low dose starts to disintegrate when dealing with radionuclides inside the body. And by the way, the corresponding author for this study was from DynCorp of Colorado, which was associated at the time with Rocky Flats, which makes me wonder why these researchers would use biomarkers to assess exposures for the Chernobyl catastrophe, but not for Rocky Flats fallout, at least not any research that I've been able to locate. Now, you don't have to have a catastrophic nuclear event to cause biomarker formation. In this final example, there were increases of childhood leukemia in children who resided within 5 kilometers downwind of the Kreml boiling water reactor in Germany. That's represented here on this map. Between 1992 and 1995, 21 people, including 7 parents of leukemic children, had blood draws examined for dicentrics. The rate of dicentrics in all 21 people near the reactor was significantly elevated and indicated ongoing exposures over the years of its operation. These findings led to the hypothesis that chronic reactor leakages had occurred. Following this study and others like it, the German government funded a countrywide study which showed that children under 5 years of age living within 5 kilometers of reactors in Germany had elevated rates of leukemia. A cascade of studies followed in other European countries showing much the same result. So in these examples, we see researchers using different biomarkers to assess real-world damage of radiation exposure to help reduce uncertainty of low and internal doses. And this is good because official radiation exposure guidelines use uncertainties in the health data to argue against additional protection from radiation when research is indicating otherwise. Use of biomarkers could help clarify these uncertainties and bring exposure standards into line with actual health damage. And it's important at this point that we review what additional health studies are actually showing. Dozens of studies show increases of childhood leukemia around nuclear facilities. Continuing exposure to radiation levels at or near natural background doses are associated with increases in childhood cancer, primarily leukemia, as we've seen around nuclear facilities, but also central nervous system cancers. And you can see this represented in these 2 graphs on this slide. Note that 225 nanosieverts per hour right here is about 197 millirems per year, and that is associated with a hazard ratio of 2 for these two diseases. These levels are also associated with subclinical impacts leading to behavioral problems and underperformance in school, particularly in math. After living with Chernobyl contamination for decades, studies indicate radiation damage may be carried and compounded across generations. And a study in the Bryansk region of Russia, inside this green circle, associates birth defects with living in areas of higher contamination. Some of these birth defects are de novo, meaning that they are appearing for the first time. We also have firsthand accounts of some birth defects in the contaminated areas that are not in Bryansk. And I'm going to read now a personal statement of Larissa Z. She's a mother from Belarus. My little daughter, she's different. When she was born, she wasn't a baby. She was a little sack, sewed up everywhere. Not a single opening, just the eyes. On the second day I watched her get operated on the second day of her life. The ones like her don't live. They die right away, but she didn't die because I loved her. In 4 years, she's had 4 operations. I won't be able to give birth again. I wouldn't dare. I'm scared. I heard the doctors talking. If we showed it on television, not a single mother would give birth. I went to church and told the minister. He said I needed to pray for my sins. But what am I guilty of? First, they wanted to evacuate our village, and then they crossed it off their lists. The government didn't have enough money. And right around then, I fell in love. I got married. I didn't know that we weren't allowed to love here. I wanted to get papers so that she'd know when she grew up, it wasn't our fault, my husband and I. It wasn't our love that was at fault. I fought for 4 years with the doctors, the bureaucrats, to finally get a paper that confirmed the connection between ionized radiation in small doses and her terrible condition. They refused me for 4 years. They kept telling me, your child is a victim of a congenital handicap. I studied my family tree. Nothing like this has ever happened in our family. Everyone lived until they were 80 or 90. The doctors said, we have instructions. We are supposed to call incidents of this type general sicknesses. In 20 or 30 years, when we have a database about Chernobyl, we'll begin connecting these to ionized radiation. But for the moment, science doesn't know enough about it. They called me crazy, laughed at me. I don't want you to print our name. Even our neighbors, even other people on our floor don't know. I'll put a dress on her and a handkerchief, and they say, your Katya is so pretty. Although many of these studies find disease increases, they often have difficulty linking the increases to ionizing radiation. Why is that? Well, often doses are believed to be too low to be causing the kinds of effects being seen. And there are some theories as to why these doses are actually causing the health impacts when, according to official estimation, they shouldn't be. Often, one of the reasons could be that the sensitivity of pregnancy and childhood and being female are not accounted for completely in the assumptions underlying radiation damage models. This is the formula that ICRP uses to convert from how radioactive something is in a unit called becquerel to the kind of damage that it causes, the unit of which is sieverts. This is their attempt to help us understand how much and what kind of biological damage comes from exposure to radiation. And this formula encompasses individual nuclides that are deposited in the body. So this would be for estimate of internal dose damage. There are also biokinetic models that describe basically where in the body a radionuclide goes and when or if it ever leaves. But the basis for these models has mostly been higher doses and mostly representative of a male adult. For low dose, dosimetry, and chronic exposure, these models are lacking. And as the intricacies of individualities of a fully functioning human system have been largely avoided in these formulas. So children are represented as little adults, and damage to children's organs is based almost exclusively on weight, not function or dysfunction over time. Adults are represented as slabs of meat. That's my characterization because these models are not reflecting the complexities of the damage. Cumulative damage is not accounted for. Neither are metabolic or hormone pathways, particularly the estrogenic pathway, which seems susceptible to action by radioactivity. Biokinetic models are not sex specific. A male model is still used for females. They are also not age dependent. In general, these formulas represent transport systems, but there are many more systems in our bodies, one of which is the reproductive system. Basically, research studies use a dose to a non-pregnant uterus to determine pregnancy damage. Damage to a number of processes during pregnancy are either poorly accounted for or not accounted for at all, including damage to sites and stem cells responsible for blood cell formation, the maternal exchange system and embryo organ tissues, and placental function. Although they are in the process of correcting for some of these shortcomings, we have a whole body of studies that have made poor assumptions about dose impacts, particularly from recurring lower doses. And unfortunately, right now, those studies have scooped the narrative about what the damage from radioactivity is without coming clean on the depth and the importance of the uncertainties on which they are based. So there are a lot of uncertainties with formulas for dose reconstruction and representations of damage. Underlying all of this is the uncertainty of how low is a low dose. Here we see an autoradiograph on this slide of a plutonium particle in lung tissue. Determining what is a low dose is an epistemic problem that extends beyond plutonium, particularly important for internally deposited radioactivity. You can get a very small dose when you divide it among all of the lung tissue here for this plutonium particle, a millirem if the particle was cleared out in a few days. But as you can see by the star-like deposition of the energy, the dose isn't being delivered to the entire lung. So what is this dose? Well, there's research and publication that's examining this. This 40 micron region, which is about 2/5 as wide as the strand of a hair. And for a 1 to 10 micron size particle, this cell could be getting a 1,000 to 10,000 gray dose per day. This would, of course, kill most nearby cells. But we also know that cells around those cells could be affected by this dose, even though they were not directly exposed. That's a phenomenon called the bystander effect. So again, I ask, how low is low? And what happens with a radionuclide like strontium-90, a bone seeker that lodges in the mineral bone of growing skeletons and doesn't leave for most of that person's life? This is why bioindicator research is so important. Our bodies should be telling us what our radiation damage is, rather than relying on incomplete and poorly assumed models. But clearing up uncertainties like this is not necessarily a popular undertaking, and biomarker research has received an inordinate amount of pushback. In a 2009 issue of Health Physics News, they claim that, quote, cytogenetic biodosimetry is a method used to examine radiation dose received by a person when physical dosimetry methods either require verification or are unavailable. And they state that physical dosimetry methods are personal dosimeters, 2 of which kind are pictured here. The digital one is more modern. So while they're regarding biomarkers as a second-tier research technique, they seem to be conveniently forgetting that people don't always wear badges or use other measuring equipment properly or at all. And even if they do, a lot of the damage caused by radiation exposure doesn't end up being measured by the badge. In the mid to late 1990s, I attended a Department of Energy meeting on the impact of radiation exposure from Chernobyl. They seemed to be most interested in the cleanup workers called liquidators. I, of course, was also interested in members of the civilian public who were exposed and who continued to live in contaminated places. The DOE representative asked me to share with him our civil society contacts in the Chernobyl area. I asked him if they were going to use any kind of biomarker assessment to determine radiation exposure, such as the dicentric test. And his response was no, that was too expensive and complicated. And my response in turn was, you're not getting any of our civil society contacts around Chernobyl. I was also certainly thinking that here we have one of the most, if not the most damaging and consequential civilian reactor catastrophe, and the people investigating its impacts are cutting corners. About 2 decades later, I was in a National Academy of Sciences meeting where a physician from Oak Ridge Associated Universities who uses biomarkers to assess exposure impact confided to me that the health physicists this person worked with were skeptical of biomarker use, but that this project would pursue biomarker use despite reluctance among the health physics community. This is physician Rebecca Smith Bindman. She gave a presentation late last year to the NAS committee convened to recommend future directions for low-dose radiation research. After her presentation, physician Andrew Feinberg, pictured here, a committee member, asked if we could be looking for particular markers, cytogenetic, epigenetic, as a way to surveil pre-malignant processes like abnormal gene expression, etc. Her answer was extremely revelatory. She said, for people investigating biologic markers of radiation damage, the problem isn't size, sample size, or cost. The problem is interest in doing the study and who would be interested in funding that kind of study. In her field of medical imaging, there was a lot of antagonism to doing this kind of study, she confided. She has collaborated on private funding to look at markers, but interestingly, and also revelatory, no organization was interested in funding biomarker studies for offspring of the pregnant women who were exposed to radiation from medical procedures. Using biomarkers where possible for lower chronic radiation doses is extremely important for existing sites of contamination. As recently as 2012, NAS claimed the use of biomarkers in epidemiologic studies of low doses was unlikely to help with low dose reconstruction, as the variability of the assays within a person or between persons was a major problem. But they did recognize, quote, rapid advances were occurring and that uncertainties in dose reconstruction models could be reduced using biomarker science, end quote. Dr. Robert Ulrich with RERF, the group of researchers that studies the atomic bomb survivors, is hoping to use biomarkers that can extend down to lower doses to investigate cardiovascular disease among that cohort. And at this point, I should remind everyone of the how low is low alpha particle slide and note that for doses from internally deposited nuclides, biomarkers may be perfectly suitable for dose reconstruction, whether or not the dose is characterized as low. Here's why understanding lower dose impacts is so very important. It took just 3 wildfires in the so-called exclusion zone of Chernobyl in the early 2000s to redistribute an estimated 8% of the original cesium-137 released in 1986. And they've had many and larger fires since. According to a Greenpeace report on Fukushima, stocks of radiocesium in forests, hill slopes, and floodplains can be remobilized and contaminate areas downstream by significant rainfall events, typhoons, and snow melt. And when trees lose their leaves, which are recycled back into the environment, the radionuclides those tree roots pull up from far down in the soil line become available again for widespread biouptake. Once contamination is out in the environment, it is a continuing threat that is extremely hard to contain. The cleanup of Rocky Flats and the restricted use as a wildlife refuge will do little to ensure that the remaining contamination there or outside the fence line will not spread or become resuspended. Consider the Marshall Fire, here represented by this salmon-colored area, and here's Rocky Flats, right? None of this is a surprise. The community around Rocky Flats has already experienced many of the health impacts I reviewed earlier from studies of other radiologically contaminated sites, leukemias and other cancers, as well as higher birth defects, et cetera. So here's a list of processes and events that can cause resuspension of radionuclides. Human activity like vehicular traffic, or most recently war, fire, wind, snow melt, leaf litter fall, decay and recirculation, rain, animal activity, soil erosion, water movement. The environment circulates, recirculates, and recycles. We should never forget that. But it's not just radioactivity at Rocky Flats. Complex environmental exposures from a variety of sources, such as chemicals, infectious agents, and lifestyle factors, and how they affect a person's health is called the exposome. And a key to understanding it is using biomarkers. The study I mentioned by Korsakov earlier in the Bryansk region of Russia uniquely also examined and demonstrated additive and potentially synergistic impacts in areas contaminated by Chernobyl radioactivity and industrial chemicals. At Rocky Flats, non-radioactive contaminants like asbestos and beryllium are also part of the contamination profile. So the Reeses and Commoner used the information they collected to start the push for the Partial Nuclear Test Ban Treaty, with one phone call from then President John F. Kennedy to Louise, and they witnessed a 50% decline in strontium-90 in children born in 1968, after the PTBT went into effect in 1963. This graph here also shows a steep decline in carbon-14, also due to the PTBT. It may well not have happened without revelatory experiments that came from a coalition of independent science, academia, activism, and organizing. But remember, even though Louise and company managed to halt nuclear testing anywhere but underground for the most part, each of us carries a bit of the cesium-137 from these atomic tests, according to the Environmental Protection Agency. And although these tests are largely relegated to history, and let's hope it stays that way, the damage that they have caused us probably isn't. So like the Reese's scientifically defensible and independent efforts to collect baby teeth and look for strontium-90, the only way to push back against the official and limited use of biomarkers is by independent biomarker studies. Supporting and doing this work puts you in good historical company. This is a list of resources on this slide that I've used to create my presentation.
Thank you so much for listening to me today. If you have any questions, I will take them.